Hello?
Okay. Yeah, hi.
Everyone. Okay, so Shanlin Wang now will have a presentation on marine sea ice biogeochemistry in E3SM model.
Okay, can you see my screen okay?
Yes, yes, we can.
Okay, great. Thank you. So today I want to talk a bit about some achievements in ongoing work from the ocean and sea ice biogeochemistry at Alamos. So first I want to start with some introduction and to point out why we need to understand and study ocean and sea ice biogeochemistry and point out the importance of this component in the Earth's model. So first of all, we know the global ocean play an important role in the global carbon cycle, and it takes up 20 to 35% of anthropogenic carbon, and also it can maintain significant amount of carbon into a deeper ocean for a very long time, and then it regulates the climate. Among all the global ocean, we know coastal ocean is also an important area to understand in terms of the carbon cycle, because 80 to 90% of organic material or sedimentation processes happen in the coastal zone. So we need to understand the ocean carbon cycle, in especially the coastal zone, how it responds to the climate and feedback. And the second reason why we want to study the ocean biogeochemistry, because the biology plays an important role in the radiation budget. So here I'm showing you some results where we compare the prognostic chlorophyll versus the climatology reconstruct based on satellite data. And so on the left, I'm showing you comparisons that we did using previous CSM code. And you can see using the dynamic chlorophyll versus the climate fixed chlorophyll concentration can create more than 1 degree difference in sea surface temperature. And this because chlorophyll can affect the radiation penetration in the upper ocean, in which affect the heat distribution. And other modeling study also suggested that chlorophyll, in which type of chlorophyll we use, whether it's dynamic chlorophyll versus the climatology, can make significant difference in the ENSO simulation. Figures on the right is from Kang et al. 2017. Sorry, I was wrong here. But basically, the authors show that using the interannual varying prognostic chlorophyll can significantly improve the ENSO simulation in CSM code. And it can improve both the frequency as well as the intensity of ENSO simulation. So this is another reason why we need to understand or simulate ocean biogeochemistry. Another reason is because marine biogeochemical activities can produce significant amount of biogenic aerosol precursors, which can affect the climate. Here, I'm showing you some previous study we did at Alamos, and to examine the impact of DMS, which is an important marine biogenic aerosol precursors. And with and without the DMS from marine biology, we see it can cause a significant difference in terms of the cloud radiative forcing, as well as the surface temperature. It can change the surface temperature simulation by over 5 degrees. And given the importance of marine BGC, we need to implement and develop the module. And for the E3SM V1, we have certain scientific goals to meet. And here is the requirement for ocean BGC. First, because we need to analyze the feedback, which require both land and marine BGC. So we also need to provide multiple CO2 tracers to allow the isolation of carbon cycle processes. And we also implemented the global CIS BGC, which is a unique capability among different Earth models. And so to meet those goals, we first imported pre-existing biogeochemical elemental cycling module into the MPAS ocean framework, which requires significant amount of efforts because we need to change the model structure and then also columnize it. Now in the ocean BGC part, I mean the ocean BGC itself, it has increased complex processes and with many different species as well as complicated processes. But right now we only considered 5 phytoplankton functional groups to represent the key biogeochemical functions in the global ocean, especially for the carbon cycle. And we also considered 1 zooplankton group, and as well as some key phytoplankton growth nutrients. In these simulations, in the current module, all the processes are super simplified, and most of the processes are prescribed or parameterized to cut the computational cost. On the CS part, Nicole implemented a global CS biogeochemistry. As I just mentioned earlier, the 3D global CS biogeochemistry is a unique capability in E3SM, which includes multiple phytoplankton functional group, as well as some key nutrients, and also the fluxes between ocean and sea ice. With the implementation of CS biogeochemistry, now we can simulate the 3D distribution of biogeochemical tracers in sea ice. And as you can see on the right, from the observations, people saw some bottom layer biogeochemical biological activities in sea ice in Arctic, in which is mainly controlled by the microscale processes, the brine dynamics, which was reasonably reproduced in the current CIS BGC code. And in the Southern Ocean is operated by different processes, which lead to a full vertical penetration of biogeochemical tracers. As you can see, we also the result is also well reproduced. And here I'm showing you the global CIS primary production. We can simulate both hemispheres at the same time. And the distributions is, we consider the distribution is reasonable, but it's very difficult to validate against any data because the sparseness of the data. But Nicole did validation against individual in situ data sets. And here you can see the upper 3 panels shows you the comparison between model simulation and observations in Arctic. And the lower 3 panels shows the result in the Southern Ocean. And different curves shows in the shade shows the model simulated result, in the dots individual observational data. And you see, we can produce reasonable seasonal cycle as well as the magnitude of chlorophyll in sea ice. And of course, there are still some remaining biases to be tested in next steps. And on the ocean side, we also validate the model against the observational data. here I'm showing you comparison between the CSM results, the previous E3SM code-based results, and the observational glow depth data, as well as the E3SM V1 simulation result, which the later one shows in the blue color. And you can see these simulations shows very good distribution, comparable distribution with the observational data. But there are still some mismatches. Here I'm showing you the zoom in into the upper ocean alone, and on the left 6 panels I showed you the result from the northern hemisphere greater than north of 40 degrees north, and on the right I'm showing you the result from source of 40 degrees source. And as you can see, although we have a nice comparison on the global scale, we still have regional compensating errors, in which require further tuning. And here I'm showing you the results of annual mean chlorophyll concentrations, and with the E3SM annual mean chlorophyll concentration in the upper panel, and the satellite observational climatology at the bottom. As you can see, we get similar patterns compared to the satellite data, although we do have biased low coastal production. And all these coastal biases are likely due to several reasons. First of all, the resolution in these simulations are too coarse to resolve all the coastal processes. And also the ocean biogeochemistry was tuned and configured for global open ocean in the past. And so we couldn't really represent the perimeter values. It's now tuned to represent the coastal processes. And also there are missing processes to resolve the coastal biogeochemistry. But because we focus on the carbon cycle in E3SM V1, we can show that RC CO2 flux are reasonable compared to observational-based estimates shown in the lower panel. And although we do see some regional biases, the biases can also be seen in the seasonal cycle. And the black curve shows the observational-based estimate in terms of seasonal cycle, and then the red dashed line shows the model simulation result. And as you can see, we have too much or too little CO2 uptake in the from February to May, and probably too much in the other half year. And also we got some biases for pre-industrial RC CO2 flux, because right now we have a weak sink in the pre-industrial CO2 flux estimates. However, other studies or other model simulations suggest a weak source for ocean during pre-industrial era. And all these biases can largely attributed to the physical state in the ocean, for example, some biases in SST, and also there are still remaining biases in biological carbon uptake. However, currently we don't have any feedback from ocean BGC to the climate, and so for V1, we still working on the simulation, and that the results shouldn't be significant issue. However, we do have concerns moving forward. And then about the next step, because the scientific, we think of some key scientific questions to ask. First one is, what are the impacts of different energy and land use futures on the ocean carbon cycle? Here, I mean both ocean and sea ice. And also, we want to understand how the marine carbon cycle and biological activities will feedback to the coupled system. The last one, we also want to ask how might coastal marine ecosystem respond to changing terrestrial and atmosphere inputs. As I mentioned earlier, coastal zone is very important in the Earth system, and as well as regulating the climate state. We did some preliminary tests and using a previous E3SM code base. In those tests, we compared the impact of CS biogeochemistry. And here, in the upper panel, shows you the contour of CS coverage. In the background color shows you the ocean surface chlorophyll in the default state. And when we consider CS biogeochemistry, we compare the CS the fully coupled CS and ocean biogeochemistry versus one-way coupled CS biogeochemistry. Now we see a significant increase in terms of the overall biological production when we consider the CS biogeochemistry. And there's also dynamic seasonal variation in terms of the cause and the contribution of CS BGC and ocean BGC. And with that said, we want to move the investigation further to climate impact from CS BGC. And to do that, first we want to enable the CO2 exchange through the CS covered area, because right now we assume there is no gas exchange in ice covered area, which is an incorrect assumption and unrealistic. So we do need to improve that. And another another issue we want to resolve is activate snow on ice biogeochemistry to address the direct impact of biogeochemistry on sea ice albedo, as well as the trace gas production. And to do that, we need to enable the direct atmosphere ice fluxes of trace gas and other constituents. And also, we want to improve the carbon sequestration estimate. For example, we know the brine rejection can bring the biogeochemical tracers variables to deeper ocean, which is not considered or properly simulated in the current code base. Then moving on to the ocean biogeochemistry part. As I mentioned earlier, although we made some achievements in the overall code implementation and testing, there are still significant biases in the biogeochemical simulations. And to solve those, to resolve those biases and fix those biases, we have a short-term plan and long-term plan to meet the V2 simulation requirement as well as beyond. So for the short-term plan, first we want to implement scale-aware parameterization to improve the regional refined BGC simulations. As I mentioned earlier, we cannot fully resolve the coastal processes, largely because the biogeochemical module is parameterized and tuned to match the observations from the open ocean. It's not set up for the coastal simulations, and we do need to improve that and change the parameterization there. And also, we want to implement supercycling of biogeochemistry time stepping to reduce the computational cost. And Matt has planned to work on this. However, it'll be great to enable this capability, which can we consider this can significantly reduce the ocean BGC computational cost. And then another short-term path is to implement two-way coupling between atmosphere and ocean, as well as land and ocean. This can allow us to diagnose the impact of anthropogenic activities on the ocean biogeochemistry, and also allow us to investigate the feedback from ocean biogeochemistry to the climate. In this, what I just mentioned was the short-term plan that we consider we can finish within the V2 timeframe. And moving forward, as I just mentioned, the ocean BGC, most of the ocean BGC processes are prescribed and parameterized in a simplified approach. And all these equations and many of the parameterization will construct based on knowledge about 15 years ago. And by now, we have learned quite some new information about ocean biogeochemistry. And we know we have key missing processes to consider. And for example, we didn't have any explicit microbial activities, which is key component in terms of the organic matter cycling. It can play an important role. This component plays an important role in transforming organic matter into inorganic form, as well as transform labile organic matter into the recalcitrant organic matter, which can be stored in the ocean for thousands of years. However, this process was costly prescribed and has no response or feedback to the temperature change or climate. And so moving forward, we consider is a key component to be implemented. And another consideration is about the organic matter export. So for the ocean carbon cycle, there are two key components. One is the solubility carbon pump. Another is the biological carbon pump. And for the biological carbon pump part, there are three important processes play in a role. One is the photosynthesis, which can produce the organic matter, convert the inorganic carbon into the organic form. And then also the cycling and recycling of organic matter in the upper ocean, where the microbial community plays an important role. Another component will be the export of organic matter to deeper ocean, which can allow the carbon to be stored in deeper ocean for much longer time scale. And however, all this export is prescribed based on a Martin curve, which was constructed over like several decades ago. And we know the equation is not realistic or physical based on the current knowledge, and it involves complex processes in the export flux, as I've shown in the figure. And so we do need to understand what is the best way to simulate and configure those export fluxes. Although all these microbial processes in export fluxes requires understanding in initial simulation of complex processes, it doesn't mean we need to keep increasing the complexity of the BGC module. We just need to understand the processes and update the parameterization or the prescription. Because right now, all the configuration, the parameterization was based on some knowledge, old knowledge, and we need to update it and also allow it to respond to temperature change as the community will understand now. Okay, so I think that's all about all I want to talk about today for the ocean and sea ice biogeochemistry. And thank you. And any questions?
Hi, Shanlin. This is Philip here. Perhaps I missed it, but what are the plans for connecting the coastal BGC to river inputs?
So this is part we mentioned for the short-term plan. We can enable the the first step would be enable the dynamic one-way coupling from river input to the coastal zone. And then we can enable the two-way coupling of nutrient flux from river input, from Mozart river input to the coastal ocean. Although we have missing processes in the ocean BGC module, and we may not be able to fully resolve all the biogeochemical cycle for those constituents from river. However, we consider first time will be enable the two-way coupling and then parameterize it to allow us to mimic the behavior.
And does Mozart have all of the nutrients and things that you need?
No, not at this moment. So right now the ocean use the input nutrient from previous simulated result by NIMS, a global model estimate. Okay, but that's the fixed climatology. And I think we can use whatever Mozart can provide us and use other constraints still from the old input files.
Hi, Shanlin, thank you. This is a really nice overview. This is Susanna. I wanted to follow up on that. I also have a question that's related to the coastal biogeochemistry, and specifically, one of the questions I am still trying to learn about is how, I know that the coastal regions have a very significant share of the CO2 flux at the ocean-atmosphere interface, but that in order for that carbon to be stored long-term, it needs to be exported probably to places where it can be stored into the deep ocean. And so I'm curious if you have a good sense for how important of a player it is in the global carbon cycle, and if you think that the model currently is capable of simulating the necessary processes for that uptake and storage from coastal regions, or if there are pieces that are missing in order to have confidence in our understanding of that.
I think what you said is largely correct. And of course, we also need to consider some local barrier of carbon in the coastal zone. And then in terms of whether we can fully resolve the carbon cycle, ocean carbon cycle, as I mentioned, we have key missing processes to represent the coastal zone. And that's why we have significant bias low biomass in the coastal ocean. Right? So in this case, we won't have confidence in reproducing the coastal carbon cycle. And that's why we do need to parameterize the reconfigure the model and parameterize better, improve the parameterization, and also implement the missing processes in the long run. However, I want to mention the coastal carbon cycle has been a key issue for all Earth system, global Earth system models. However, E3SM is probably the best model which can resolve this transport from coastal zone to deeper ocean because of the regional refined capability. So I do consider we can better resolve the coastal zone than many other global biogeochemistry module, as well as reproducing and simulate the transport. So did I answer your question?
Yeah, great. That's very helpful. Thank you.
Okay, we can also open for more questions on SCM for Peter. So any more questions? Okay, I think that we're done then. Thank you very much to both speakers. Excellent presentation. Thank you.